You're walking down a quiet street when a car backfires, your heart pounds, your muscles tense. In that instant, your brain retrieves a memory you didn't consciously choose to access. For over a century, we believed one type of brain cell controlled everything: your fears, your memories, your reactions. We were only looking at half the story. This is the psychology of people, and today we're exploring a discovery that could change everything about how we understand and treat fear itself. In February 2026, a study published in Nature overturned 150 years of scientific assumptions. The cells that researchers had dismissed as nerve glue weren't glue at all. They were architects, engineers of fear, and they've been operating in the shadows of our brains this whole time, shaping what scares us and when. Here's the part that changes everything. These cells don't just create fear memories. They help erase them, and that fact could transform how we treat PTSD, anxiety, and phobias. Let's go back to the 1850s. A German pathologist named Rudolf Virchow is peering through his microscope at brain tissue. He sees neurons. The cells we now know carry electrical signals. But between those neurons, he notices something else. A dense matrix of other cells, smaller, star-shaped. He doesn't know what they do, so he calls them neuroglia, nerve glue, and that name stuck. For more than 150 years, neuroscientists focused almost exclusively on neurons. The other cells, the astrocytes, were treated as mere scaffolding, background machinery. But here's a number that might surprise you. Astrocytes actually outnumber neurons in the human brain. The support staff outnumbers the main characters. That fact alone should have raised questions. The paradigm started shifting in the 2000s. Researchers began noticing that astrocytes weren't just sitting there. They seemed to be communicating, signaling, doing something. But doing what exactly? That question lingered for decades. The technology to answer it simply didn't exist yet. We could see activity, but couldn't prove causation. Then came February 2026. A research team published their findings in Nature, and what they discovered wasn't subtle. It was a complete rewrite of our understanding. Using fluorescent sensors, researchers tracked astrocyte activity in real time. They focused on the basolateral amygdala, the brain's fear processing center, and watched what happened during learning. What they observed changed everything. When fear memories were being formed, astrocyte activity didn't just stay stable, it increased dramatically. These weren't passive bystanders. And here's where it gets really interesting. During fear memory recall, when the brain retrieved a scary memory, astrocyte activity spiked again. They weren't just building memories, they were fetching them. But the most significant finding came during extinction learning. When animals learned that a previously scary stimulus was now safe, astrocyte activity declined. They were helping unlearn the fear. The researchers didn't stop at observation. They designed experiments to test causation. What happens when you deliberately increase astrocyte signaling or suppress it? When they selectively increased astrocyte signals to neighboring neurons, something remarkable happened. The strength of fear memories increased in parallel. Stronger signals, stronger fear. And when they suppressed astrocyte activity, the fear memories weakened. The relationship was direct, causal, undeniable. Astrocytes weren't just correlated with fear, they were shaping it. But the story doesn't end in the amygdala. The researchers discovered that astrocyte manipulations affected how fear signals were relayed to the prefrontal cortex, your brain's decision-making center. This means astrocytes influence not just what you fear, but how you respond to that fear, whether you freeze, flee, or recognize the threat has passed. The executive decision, as the study's authors put it, and I think this quote is worth sitting with, astrocytes are not just passive bystanders, but active participants in shaping fear. Now, let me put this in perspective. Every current treatment for PTSD, for phobias, for anxiety disorders, targets neurons. Every medication, every therapy protocol, neurons only. We've been working with half the cellular machinery. It's like trying to tune a piano by only adjusting the white keys, while ignoring the black ones entirely. Current PTSD treatments work for many people: exposure therapy, cognitive behavioral therapy, certain medications. They help, but the relapse rate remains significant. What if, and this is the question the researchers are now asking, what if targeting astrocytes could enhance extinction learning? Could help people unlearn fear responses more completely? The therapeutic implications are significant. An entirely untapped cellular system now presents itself as a potential intervention point. A new door has opened. But I want to be careful here, because it's easy to oversimplify, and that wouldn't serve you well. Let's talk about what we don't know yet. Astrocytes serve many vital functions beyond fear memory. They regulate blood flow. They clean up waste. They maintain the blood-brain barrier. They're essential infrastructure. Any treatment targeting astrocytes would need to be incredibly specific. A hammer won't work here. This would require a scalpel, and we haven't built that scalpel yet. Some neuroscientists caution against overstating the clinical implications. This is fundamental research, important, groundbreaking, but early stage. Translation to human treatments takes years. The research was conducted in animal models. Mice. The leap to human brains, with their vastly greater complexity, isn't automatic. It requires careful, methodical work. So, where does this leave us? What can we actually take away from this discovery today? Not 5 years from now, but right now. First, this research may help explain why extinction-based therapies actually work. Exposure therapy, fear extinction protocols. They engage cellular processes at multiple levels, not just neuronal ones. If you've tried exposure therapy and it helped, even partially, this research suggests your brain's astrocytes were part of that success. You engaged a system you didn't even know existed. Second, this discovery suggests that fear memories are more dynamic than we previously thought. They're not fixed recordings, they're actively maintained, and can be actively modified. For those living with anxiety disorders or PTSD, this might be worth considering. The fact that cellular systems exist specifically for fear extinction suggests your brain has built-in mechanisms for recovery. These mechanisms can be supported. Sleep, which plays a role in memory consolidation, consistent therapeutic work, and patience with the process. Cellular change happens, but it takes time. Third, and this is the broader takeaway, the brain's support systems are vastly more sophisticated than we knew. What else might we be underestimating? This pattern has repeated throughout the history of neuroscience. Structures dismissed as unimportant later reveal critical functions. The brain keeps surprising us. The glymphatic system, discovered only in 2012, revealed that the brain has its own waste clearance system that activates during sleep. Another support function hiding in plain sight. So what happens now? The research team is already planning follow-up studies. They want to understand the molecular mechanisms, exactly how astrocytes communicate with neurons during fear processing. Eventually, and this could take years, the goal is to identify specific molecular targets, proteins or receptors on astrocytes that could be influenced by drugs designed specifically for fear extinction. But even before treatments emerge, this discovery shifts our fundamental understanding. Fear isn't just a neuronal phenomenon, it's a collaborative process between multiple cell types working together. If you're interested in this kind of neuroscience research and want to stay updated, consider subscribing to this channel. We explore discoveries like this every week, always with the nuance the science deserves. Now, I want to return to something we mentioned earlier, the history of how astrocytes were named and what that name revealed about scientific assumptions. When Rudolf Virchow called them nerve glue, he wasn't being dismissive. He was being honest. He didn't know their function. But that label shaped perception for over a century. The lesson here extends beyond neuroscience. What we name things shapes what questions we ask about them. Support cells don't invite investigation. Active participants do. This study is a reminder that settled science sometimes isn't as settled as we think. The frontier of discovery often hides in things we stopped questioning. So, here's what I'm left thinking about. For 150 years, we studied the obvious actors, the neurons, the electrical signals, the dramatic flashing of brain activity. Meanwhile, in the background, outnumbering the stars of the show, the astrocytes were doing essential work, shaping fear, enabling memory, helping us learn when it's safe to let go. What else might be operating in the background of your brain? Or your life, shaping things in ways you haven't considered? That's worth sitting with. I'm thinking about it too. This has been the psychology of people. If this changed how you think about fear and memory, even a little, that's exactly what we're here for. Until next time.